Hi everyone, this is Rima, your biology educator from V-Learning. So as you know that V-Learning is an online educational platform where we provide quality education for all. So to get all the benefits from our channel, do subscribe to it, and also don't forget to visit our website, that is www.v-learning.in, where you will be getting all the pre-recorded videos, the live classes, animated videos, and also the PDF notes of all these lectures. So let's begin today's session. So what you are going to learn in today's class, you remember that in our last class we have completed chapter number 12, right, which deals with mineral nutrition in plants, right? So today we are going to start with your chapter number 13, which deals with what? Which deals with photosynthesis in higher plants, right? You all studied about photosynthesis from your lower grades, isn't it? So how does the photosynthesis takes place? Okay. So when photosynthesis takes place, what are the various reactions taking place during the photosynthesis? How the electrons are being transferred in the chlorophyll, in the chloroplast. Okay, so you will be learning that what are the various pigments, then what are the various light reaction, what are the various dark reaction taking place in during the process of photosynthesis in higher class in higher plants in this chapter. Okay, so let's begin today's class, and this is your lecture number 1. So yes, first of all, what do you mean by this photosynthesis? Okay, photosynthesis is a process by which the plants prepare their own food, right? The plants prepare their own food by using what? By using the light energy, isn't it? So that energy is being required by the plants to prepare the food in the form of sucrose, and that is being what? That is being taken by all the human or the other animals on this planet. So each and every living organism depends on plants for its food and survival. So plants are the primary source of energy, isn't it? So see, what is this photosynthesis? Photosynthesis is what? It is a physiochemical process by which plants use light energy, okay, to drive the synthesis of organic compounds. Okay, so here the plant use the light energy, and it is a what? It is a physiochemical process. So photosynthesis is important due to two reasons. What are the two reasons? First of all, is it is the primary source. Okay, it is the primary source for all food on the earth. Okay, and it is also responsible for the release of oxygen in the atmosphere by the green plants. So without oxygen, can we survive? We cannot survive without oxygen, right? So oxygen, from where we are getting? We get it from the plants, right? We get it from the plants. So they release large amount of oxygen. So this is photosynthesis. Next, let us talk about the various early experiments which were done by the scientists to find out that what are the various components of the photosynthesis. Okay, so there were various scientists who has worked out and who found that the oxygen, then light, then chlorophyll. Okay, so they have discovered all these things. Okay, they have found that these are the essential components which are necessary for the process of photosynthesis. So one of the scientists, okay, first scientist here you are going to study is the Priestley. Okay, that is Joseph Priestley. Okay, that is called as Priestley's experiment, where he has done the bell jar experiment. Okay, he has used the bell jar, and he has used candle, he has used mice, and a mint plant, as you can see here in this picture. Okay, he has used what are the things? Candle, mice, then mint plant, and a bell jar. 4 requirements he needed. Okay, now what he has done is in 1770, okay, in 1770 he performed a series of experiments which revealed that oxygen is very much essential. Okay, essential, that is role of air in the growth of green plants, and he discovered oxygen in the year 1774. Okay, so when he has discovered oxygen, Priestley has discovered what? He has discovered oxygen. Okay, he has discovered oxygen in the year 1774, right? Now see what he has done. Okay, so what are the things he took is he took candle, then bell jar. Okay, he took bell jar, then he took mint plant, and what he took? He took candle. Okay, Now what he has done is, in his first experiment, okay, in his first experiment, what you have seen that here he has kept what? He has kept one mice and the candle, okay, and here you have seen that along with the mice and the candle, he has kept one plant too, right? So he has done the experiment, he has taken the bulb, that is the bell jar, okay. So here he has kept the mice, okay, and the candle, all right, and the candle, okay. Now after a few minutes, okay, after a few time, he has seen that the candle, it was not burning, right? It was not burning. Now, what does it mean? What does it mean? Okay, here you can see that the candle is burning, means that mint plant is releasing O2 here, right? Which is very, very much essential for the burning of the candle and survival of the mice. Clear with this? So, the mint plant is releasing oxygen, which is very, very much essential for the survival of the mice and also the candle. Is that clear? So thus he discovered that what oxygen is necessary for photosynthesis. Okay. Then next, another scientist after him, what he's done, his name is what? His name is Jen Ingenhouse. Okay, Jen Ingenhouse. So he showed that sunlight is essential. So what he has done, he has taken the plant hydrilla. Okay, he has taken plant hydrilla. So he has seen that when these plants are facing the sunlight, okay, when these plants are facing sunlight or kept in sunlight, okay, kept in sunlight, so the bubbles were coming out, from those plants, whereas when they were kept in dark, okay, when these hydrilla plants were kept in dark, no bubbles were found, okay, no bubbles were found in those plants, okay. Now from here, what he has explained, that he showed that bright sunlight, okay, bright sunlight, that is what? That is sunlight, okay. Sunlight is essential for what? Sunlight is essential for the growth of the plant, okay. Sunlight is essential for the growth and the bubble formation in the plants, whereas when the plant is kept in the dark, it will be dying, okay. It will not form bubbles. Is that clear? Yes. Next, another scientist, okay, Julius von Such. Now, what he has done is, he said that not only oxygen and sunlight, the plant in the leaves, they have got what? They have chloroplast, okay. So he has used what? He has used the he has seen, or he has examined, and he has said that the bubble formation, where it takes place? It takes place in the chloroplast, okay. The bubble formation takes place in the chloroplast, which is the kitchen of the cell, okay. What is that? It is the kitchen of the cell. So this is the kitchen of the cell, and where food is also prepared and stored in the form of starch, all right, where food is prepared and stored in the form of starch. Clear? So this was given by whom? This was given by the Julius von Such. Okay, this was given by Julius von Such. Now another scientist, that is Engelmann. Okay, now Engelmann, what he has used, he has discovered that the maximum wavelength at which the photosynthesis is taking place, that is the blue and the red wavelength. Okay. Now see what he has used. He has used algae. Okay, what he has used? He used algae, that is cladophora. Okay, cladophora. All right. Now what he has done is, using a prism. Okay, using a prism, he split light into its spectral components and then illuminated a green algae, cladophora, placed in a suspension of aerobic bacteria. Now what it took is he took a prism. Okay, now here a white light. Okay, this is a white light. All right, now he took a prism. Okay, and he has used prism, so he has seen the 7 colors coming out. You know, right? You have studied this in physics, isn't it? So splitting of 7 colors took place. Vib Geor, isn't it? Vib Geor, right? So he has observed that maximum absorption it took place at where? At the blue and the red light. Okay, blue and the red light of the split spectrum. Okay, they have the maximum capacity to absorb. Okay, so bacteria accumulated mainly in this region, right? So first action spectrum of photosynthesis was thus described. So it resembles roughly the absorption spectrum of or spectra of chlorophyll A and chlorophyll B. So this is the scientist who has explained that at what region, okay, at what region of the light reaction is taking place more, right? So next, another scientist, his name is Neil, okay, Niel, okay. So Cornelius Ventlin. So what he has done is he has used purple and green bacteria, and he was the first person to say that oxygen, okay, oxygen is evolved from the water which is being used, okay, which is being used. So what he has done, he based on his studies on purple and green bacteria. So what bacteria he has used for his study? That is purple and green bacteria. Okay, purple and green bacteria he has used, and he has demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. Okay, he has said that photosynthesis is what? It is a light dependent reaction. So in green plants, H2 is the hydrogen donor and is oxidized into O2. Okay, so from hydrogen, O2 is being evolved, okay, not from what? Not from carbon dioxide. So he has proved this one, right? Next, let us come to the kitchen of the cell, that is the chloroplast. Okay, chloroplast is the place where the photosynthesis is taking place, where the light reaction is taking place. Okay, now see, you have already studied the structure of chloroplast, so let me tell you that. So you can see this is the outer membrane, right? Chloroplast is a double layered, it is a double layered membrane, right? So it has got two membrane, outer and inner membrane, right? So this is the outer membrane, the yellow portion you can see, outer membrane. Then this is the inner membrane, okay? Now inside the inner membrane, you can see all these are the matrix, okay, where the photosynthesis is taking place. So these are what? These are the thylakoids, okay? These are the thylakoids, okay, which are the packets of energy where the light reaction is taking place. So thylakoid is the place where the light reaction takes place, okay? Light reaction is taking place in the thylakoid, okay, and they are called as grana, okay. These thylakoids together are called as what? Grana. So this region, what you can see, these are all the matrix where we call it as stroma, okay. This matrix is called as stroma, and this, what you can see, dot like structure, it is called as ribosome, right? So they have got 70s ribosomes, okay, they have 70s ribosomes, and also they have starch granules, that is the stored food, right? The stored food is what? The stored food is the starch, right? So yes, one more thing you can see, the thylakoids are joined together, right? The thylakoids are joined together, isn't it? Thylakoids are joined together with the help of stroma lamella. Okay, they are joined together with the help of stroma lamella. Now, what happens in the chloroplast? In chloroplast, the light reaction is taking place. Light reaction is taking place. And in the light reaction, okay, the photosystem 1 and photosystem 2 is being involved. Here we will be studying about photosystem PS1 and PS2. PS means what? PS is equals to photo system, okay, photosystem. So here you will see that how the electrons are being transported, okay, how the electrons are being transported from chloroplast A to chloroplast B, okay. Now let us see the various pigments, okay. The various pigments involved in the process of photosynthesis are, you will see that some of the plants are yellow in color, some are green in color, isn't it? Why? It is because of the pigments, okay. Like chlorophyll A. Chlorophyll A will be giving what? Will be giving bright or blue-green color, okay? So, chlorophyll A gives bright or blue-green color. Next, chlorophyll B, it gives yellow-green color. Next, xanthophylls, it gives yellow color, okay? Then, carotenoids give yellow to yellow-orange color, okay? Then this pigments, we can determine it through an experiment that is known as what? That is known as paper chromatography, okay? Paper chromatography. With the help of this paper chromatography, we can determine what? We can determine the various pigments, okay? The pigments in the leaf, right? Yes. Now, let us move into the light reaction. Today's class, we will be learning about light reaction, okay? That is photosystem 1 and photosystem 2 is involved in this, okay? So, here you will be looking into that what are the various cyclic and non-cyclic photophosphorylation reaction, okay? Now, see what happens in light reaction. Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates, that is ATP and NADPH. So here in the light reaction, what will happen? Here the water splitting will take place, light absorption will take place, then oxygen will release and it will result in the formation of various molecules, okay, ATP and NADPH, okay. So yes, these are required by the cell, okay. ATP and NADPH is providing what? Energy, okay. So several protein complex, here you will find several protein complex are involved, like the cytochrome, the plastoquinone, plastocyanine, okay. So these are the protein complex to which the electrons are being transported, okay. So the pigments are organized into two discrete photochemical light harvesting complexes. So that is photosystem 1 and photosystem 2, isn't it? So see here the picture, these are what? These are the pigments, okay, and this is the light energy which is being received by the pigment, okay. So this is the primary acceptor. So electron will be what? Electron will be getting excited, okay, and it will be received by the primary acceptor. So this is the reaction center. Basically the photosystems are the reaction center, okay, that will be absorbing the light and the electrons will get excited and it will be traveling with the help of what? With the help of the various protein complexes, okay. So see here what happens. The light harvesting channel, yes, LHS, right? The light harvesting complex, sorry, LHC is what? LHC is light harvesting complex, okay? Light harvesting complex. So what are the various light harvesting complex? The various light harvesting complex we will learn here is photosystem 1 and photosystem 2. These are the two light harvesting complex, okay. Light will be captured by the help of this system, okay. So the light harvesting complex are made up of hundreds of pigment molecules bound to proteins, okay. So they are bound to what? They are bound to proteins. So each photosystem has all the pigments except one molecule of chlorophyll A, okay. So forming a light harvesting system called antenna, okay. So they form a light harvesting system which is called as what? Which is called as antenna, okay, where all the light is being harvested, okay. So this pigments helps to make photosynthesis more efficient by absorbing the light at different wavelengths. Now let me tell you here, okay, there will be two wavelengths. Photosystem 2 will be absorbing the light at 680 nanometer, okay, 680 nanometer, whereas the photosystem 1 will be absorbing the light at P700, okay, at 700 nanometer. Okay, these are the wavelengths. These are nothing but these are the wavelengths in which the light is being harvested. All right. Next, so see, the single chlorophyll A molecule forms the reaction center. Okay, so here, as soon as the light comes and it gets attracted to the chlorophyll A, okay, or the photosystem, it will form what? It will form the reaction center. Okay, so the reaction center is different in both the photosystem. Okay, so in both the photosystem, the reaction center will be different. One will be having chlorophyll A, another will be having chlorophyll B. Okay, so in photosystem 1, the reaction center is chlorophyll A. The reaction center is what? Chlorophyll A in photosystem 1. And the absorption peak is what? 700 nanometer, which I have said, right? So the reaction center, okay, let's talk about photosystem 1. So the reaction center in photosystem 1 is what? In the reaction center is chlorophyll A, and the wavelength number 2, you should remember the wavelength is what? The wavelength is P700 nanometer, okay, whereas in photosystem 2, okay, the absorption maximum is 680 nanometer and it is called as P680. Clear? Yes. So in photosystem 2, okay, photosystem 2, the reaction center is chlorophyll B, whereas what happens, the wavelength, okay, wavelength is P680 nanometer. All right? Yes. Now, let us see the Z scheme of light reaction. So here you will be finding various terms. So see here light, okay, sunlight, okay, this is sunlight. So it is being absorbed by the light harvesting complex, okay. So here the photosystems will be there, okay. Now the electrons will be what? Electrons will get excited, and here this is the primary electron acceptor, okay. So this is also known as non-cyclic, okay, non-cyclic, what? Non-cyclic photophosphorylation, okay, non-cyclic photophosphorylation. The Z scheme is also known as non-cyclic photophosphorylation reaction. Okay, so here what happens, both the photosystem is involved, photosystem 1 and photosystem 2. So see here, the electron will get excited and through the electron transport chain, it will be again received by the photosystem 1. Okay, photosystem 1. It starts from photosystem 2. Then here again the electron will be excited and here the NADPH molecule will be evolved. Okay, so let us understand it. Okay, first here you should remember some terms I will discuss. Okay, so PQ. Okay, what is the meaning of PQ? Plastoquinone. Okay, so these are what? These are the protein complexes, okay, through which the electrons will be transported. Okay, these are what? These are the electron transport chain, you can say. Okay, so these are the electron transport chain. Okay, these are the electron transport chain, ETC I'm writing. Okay, then PC is plastocyanine. Okay, then you will come across cytochrome. Okay, CYT is cytochrome. So see here what happens is, okay, let's move here to the non-cyclic photophosphorylation. So here this is what? This is chlorophyll. This is chlorophyll B. Okay, these are the external apparatus of the chlorophyll. Okay, this is what? This is chlorophyll. This is chlorophyll A. Okay, this is chlorophyll B. Sorry, This is chlorophyll B. As you have seen that the reaction center of photosystem 1 is chlorophyll A, right? So here it will be chlorophyll B. The reaction center is chlorophyll B. So this is what this is, photosystem 2. Okay, this is photosystem 2. That means P680. Okay, P680. Okay, so the reaction center will be P680. Now this is what, this is the sunlight. Okay, this is the sunlight. So the light will be coming and being absorbed here. Okay, and being absorbed in the chlorophyll. Now here there will be primary electron primary electron acceptor. Primary electron acceptor. Now see what happens is the electron, okay, will get excited from here. The electron will get excited. So two electron will get excited and will be accepted by the primary electron acceptor. Okay. Now here what happens, the various series of reaction, that is electron is again transported to the various protein complexes. So here plasto PQ, plastoquinone, then what is present, cytochrome, then next plastocyanine is present here, right? So again this electron are being sent to this through this electron transport channel. Okay, now it will be received by whom? It will be received by the photosystem 1. Okay, so photosystem 1, the reaction center is what? The reaction center is chlorophyll A. Okay, the reaction center is chlorophyll A. Okay, so this is what, this is photosystem 1. Okay, which has a nanometer of P700. Okay, so the wavelength of photosystem 1 is P700. So the electrons will be received here, right? Again it will be excited. Okay, so again it will be excited and it will be sent to the primary electron acceptor. Okay, again here is a primary electron acceptor. So this electron, okay, this electron now will be sent to where? To the ferridoxine. Okay, one more thing I should write here. FD means what? FD means ferridoxine. Okay, FD means ferridoxine. All right, now see here what happens during this electron transportation. What happens here in the cytochrome, one molecule of ATP is being released. Okay, so ADP leads to the breakdown of or release one ATP molecule here. Okay, one ATP molecule here. And here in the outer membrane, what happens is from NAD, okay, NADP plus, from NADP plus, okay, what is released? What is being formed? NADPH, NADPH molecule is being formed. So here, as you can see, it's one molecule of ATP, and one NADPH is being formed during this non-cyclic photophosphorylation, right? One molecule of ATP and one molecule of NADPH is being formed. Now, see here what happens, all the electrons are being transported. It is not coming back. It is not returning back here to the photosystem 2. Now what happens to the photosystem 2? Here again what happens, the photolysis reaction is taking place. So here the photolysis reaction takes place in which the water H2O is splitting. Okay, splitting of water is taking place. So water gets splitted into water, and this is called as photolysis. Okay, photolysis, where splitting of water is being taking place in order to send back the electron to the reaction center, that is photosystem 2. Okay, so see here what happens. So H2O, it splits and gives rise to two H plus 1 nascent oxygen plus 2 electron molecule, right? Now this electron, it will go back here in the photosystem 2. Is that clear? This electron is going back in the photosystem 2, right? Now it is getting the electron. What happens to the oxygen? The oxygen will go back to the air, right? The oxygen will go back to the air, isn't it? So this is how the non-cyclic photophosphorylation is. So what happens in this reaction? As you can see that it involves two photosystem, right? Non-cyclic photophosphorylation is a Z scheme. Okay, so see, this looks like a Z, right? So it's a Z scheme, and here two photosystem are involved, photosystem 1 and photosystem 2, right? Both the photosystems are involved here, isn't it? And here there is there is production of what? One molecule of ATP and one molecule of NADPH, right? NADPH, isn't it? And there is photolysis of water also, right? Photolysis of water also, and oxygen also is being evolved here. So you can see oxygen is also being evolved, right? So this is non-cyclic photophosphorylation. Okay, so yes, next move into the cyclic photophosphorylation. So these are all the light reaction which is taking place in the chloroplast, okay, which is taking place in the chloroplast. So here what happens, that is cyclic means the electrons are returning back. Okay, so see here, this is the light. Okay, then chlorophyll, only photosystem 1 is being involved in the what, in the cyclic. Okay, only photosystem 1 is being involved. Okay, P700 nanometer is the wavelength of photosystem 1. So the electrons will get excited. Okay, it will reach to the primary electron acceptor. Okay, and again it will return back. Okay, the electrons will be transported through the electron transport system. Okay, and it will again return back to the photosystem 1. So let's see how it works. Okay, and in that process, ATP leads to the formation, ADP leads to the formation of ATP. One ATP molecule is being formed. So see, cyclic photophosphorylation, here there is a light. Okay, that is sunlight. Now the light energy will be harvested by the chlorophyll, isn't it? That is only here what is involved. The reaction center is chlorophyll A. Here only photosystem 1. Okay, photosystem 1 is being involved. What is the wavelength? It is P700, right? P700 is the wavelength of photosystem 1. So the light will be harvested here. Now it will be accepted by the primary electron acceptor. Okay, but the primary electron acceptor. Now here what happens is the electrons are getting excited. Okay, the electrons will get excited to electron, right? Now what happens through the electron transport channel? Okay, ferredoxin. Here is the ferredoxin. After the ferredoxin is the plastoquinone. Then after the plastoquinone is the cytochrome. Then after the cytochrome is the plastocyanine, right? Plastocyanine. Now during this process, what happens? One ATP molecule is being formed from ADP. So one ATP is being evolved from ADP, right? But there is no evolved of oxygen. Oxygen is not evolved, and photolysis is also not taking place, isn't it? Only one photosystem is involved here, right? So the electrons will again come back to the photosystem 1, right? So this is the cyclic photophosphorylation. Okay, this is cyclic photophosphorylation, where what you will find? Where you will find no photolysis. No photolysis. SIS. Okay. So yes, so see, this is the difference. You can write it down. Only photosystem 1 is involved, right? And here both the photosystems are involved, photosystem 1 and photosystem 2, right? So no photolysis of water is taking place. No photolysis, you have seen here, like water is not split. Here water is being splitting. Photolysis is taking place, right? That is, water is being split into two hydrogen plus oxygen as in oxygen and 2 electron, right? That is splitting of water is taking place. Then here you have seen that oxygen is not evolved, isn't it? Here oxygen is evolved in the non-cyclic photophosphorylation. Then NADPH plus, H plus is not synthesized. Only ATP is being synthesized in the photosystem 1, right? But here in the photosystem 2, NADPH and H plus is synthesized, right? Next, what happens is what electron is cycled back to the reaction center. As you can see here, the electron is coming back to the reaction center or not. The electrons comes back to the reaction center. But here, that is in the non-cyclic, the electrons will not come back to the reaction center. So this is very, very easy, right? Simple. So yes. Next, let us move into the chemo-osmotic hypothesis. Okay, now what is this chemo-osmotic hypothesis? So see, this is the diagram, which is the diagram of the chloroplast only, okay? That is where the reaction is being taking place. So this is what, this is the thylakoid, okay? This is the thylakoid membrane. These are the photosystem, okay, and these are the electron transport system, okay, cytochromes. And this is the lumen and this is ATP synthesis. Now what happens here, let us see, this is the stroma, okay. Now, during the process of that is in the non-cyclic photophosphorylation, you have seen that photolysis is taking place, okay, photolysis is taking place. So what happens during this process? Because of this photolysis, lots of hydrogen gets accumulated into the lumen, okay. So this hydrogen makes the lumen acidic, so it needs to be thrown outside the cell, okay. Now what happens, look here, is this is what, so this is the thylakoid membrane, okay. Here will be the photosystem 2, okay. Now here is the photosystem 1. Now see here, this is the thylakoid membrane where the light reaction takes place, okay. Thylakoid membrane where the light reaction takes place. And here there will be the ATP synthetase, okay. There will be ATP synthetase enzyme, okay, ATP synthetase enzyme. Now, as you know that in the photosystem 2, what happens? Photolysis takes place, right? So it takes place in the lumen, okay, it takes place in the lumen. This is the lumen, and these are all, this area is the stroma, okay. This area is what? This area is the stroma, as you can see here, right? So this area, these are all the stroma of chloroplast, okay. These are the stroma of chloroplast, okay. So this is all taking place in the chloroplast only, okay. So see here, so this is having F0 and F1 particle, okay, F0 and F1 particle, okay. So as you can see here now, so what happens basically during the process of photolysis in the photosystem 2, right? In the photosystem 2, during the process of photolysis, the hydrogen is splitting of water is taking place, right? Splitted into what? 2 hydrogen plus oxygen and 2 electron, isn't it? So because of which lots of hydrogen gets accumulated here in the lumen, okay. Lots of hydrogen will get accumulated in the lumen and it will make the lumen acidic. It will make the lumen acidic. Is that clear? It will make the lumen acidic. Now, this hydrogen ions needs to be thrown out of the cell, okay, needs to be thrown out. Now, through this ATP synthesis, it is through this protein, okay, this is nothing but these are the spatial proteins, okay. These are the spatial proteins, okay, F1 and F0. These are the spatial proteins which helps in the removal of these hydrogen ions from the lumen, okay. So, this will take out the hydrogen ions from the lumen, okay. And it uses what? It uses this enzyme, that is ATP synthetase, where ADP is converted from one ATP is being formed, okay. So during this reaction, what happens? From the ADP, one ATP molecule is being formed. Is that clear? During this process, what happens? from one ADP molecule, one ATP is being synthesized. So this is how the ATP is synthesized through the chemiosmosis hypothesis or chemiosmosis. Okay, so this is a hypothesis which is given by one scientist. Okay, so see, this ATP is very much necessary for the cell to get the energy. This is the process how the ATP is being synthesized after the electron transport chain. These are all the electron transport chain which you have studied here. In case of plants, this is the process taking place. Okay, so here how the from the ATP the energy we get it. Okay, so this is all about it what you have studied in today's session. Okay, so you can write down that chemoosmosis requires a membrane. Yes, it requires a membrane. Okay, that is the thylakoid membrane. And also what it requires the proton pump. Okay, it requires the proton pump. These are the special pumps which are pushing the hydrogen ions outside, isn't it? So it requires proton pump. So what these proton pumps do, they collect all the hydrogen ions and they act like a motor. Okay, they revolves, and in that case they produce one ATP from ADP. Okay, so see here, energy is used to pump the proteins across the membrane to create a gradient or high concentration of proteins within the thylakoid lumen. So here what happens, energy, huge amount of energy is being required. Okay, so ATP synthase has a channel that allows the diffusion of the proteins or protons back across the membrane. This releases enough energy to activate the ATP synthesis enzyme that catalyzes the formation of ATP. So here what happens, to remove this hydrogen ions, it requires lots of energy. Okay, so this enzyme, with the help of this enzyme and with the help of this pump, okay, this is a proton pump. This is a proton pump. With the help of this proton pump, okay, so and with the help of this ATP synthesis, all the ions are being removed from the lumen. Okay, and it requires energy. So in that case what happens, it leads to the formation of one ATP molecule. Is that clear? This is chemiosmotic hypothesis. Okay, so in today's class we have learned about what we have learned about the light reaction in the plants. Okay, how the light reaction is taking place in the photosynthesis process. In our next class we will be covering the dark reaction in the plants, where you will be learning about the C3 and C4 pathway. So I hope you have understood what is being discussed in today's session. So if you find this video useful, do like it and also share it with your friends. So I will meet you in my next class, where we'll be learning about the dark reactions. So till then, take care and bye-bye.